Guten Tag. Today we're going to learn about translation, which is the next step that follows after RNA is made. So we've talked about DNA replication, turning DNA into more DNA. We talked about transcription, which is turning DNA into RNA. Now we're going to talk about translation, which is turning RNA into proteins. Now, just like translating something from one language to another is taking the same information and put it in a different way, we're going to do the same thing. We're going to take the information that's in the RNA and we're going to put it into a protein. So translation is the process of using the information coded on a strand of mRNA to make proteins. So this right here is a strand of amino acids. There's an amino acid, there's an amino acid, and so on. And what happens is these amino acids start to kind of curl up, very similar to DNA. And those little curls you can see in here start to form complex shapes. And then they, here it is right here, that thing, curls up with even more to make these big complex structures. This particular structure right here is hemoglobin, which is what's responsible for carrying oxygen throughout your blood. In order for translation to occur, you need four things. One, you need mRNA because he's got the message. Two, you need tRNA because he carries the amino acids to the party. Three, you need rRNA because that's the ribosome where proteins are made. And four, you're going to need a whole load of amino acids. Where do those amino acids come from? The food you eat. So when you eat proteins, you break them down into the individual amino acids, which go out into the cell cytoplasm for you to reassemble into your very own proteins. Okay, before we get started on the actual process of how this occurs, we need a little bit of vocab. So the first thing we need to do is talk about the genetic code. So you've probably heard about this before. It's the sequence of mRNA nucleotides that tells us what amino acid to use. So the genetic code is a way to relate the information on the mRNA to a corresponding amino acid. It's kind of like maybe like a Spanish dictionary. So when you have a word in Spanish, hola, you look up hola in the dictionary and it says, oh, hola means hello. Okay, well, same kind of thing. So if I have a chunk of nucleotides, AUG, well, that's my word that I don't understand. So I use this guy over here in my dictionary to figure out what that word means. And so what we're going to learn is that the nucleotides come in chunks of threes called codons. And this is how I read those codons. I start with the very first one, A. So I go over to the first one, A. The second one is U, so I look up here, U. And the third one is G, over here is the third position. So now you can see that these two intersect here and here. So this box right here. Now I have the first and second one here. The third one we said was G, so G means it's going to be this one right here. So what that means is that AUG means methionine, which is an amino acid. So just like hola means hello, AUG means methionine. So the genetic code is kind of like my translating dictionary. Now we can see in here all the different names of the amino acids in there. No, you don't have to memorize them. You will be provided this chart, but you do need to know how to use it. Now in here we have 20 different amino acids. If you remember that from chapter 2, that there are 20 different amino acids that I can rearrange in a variety of different ways to make different proteins. And so that's what all these amino acids are right here. Now, I need to read on my mRNA, like I said, a group of 3 amino acids. I'm sorry, let me try that again. A group of 3 nucleotides. A group of 3 nucleotides is called a codon. And you can kind of think of it, the fact that it codes for a particular amino acid. So it's a series of 3 nucleotides found on the mRNA. That's kind of important there. And it codes for a particular amino acid. And so if I have an mRNA strand and it says, AAU, GAU, UAC. Well, every 3 of these equals 1 amino acid. So AAU, I would go A, and U, asparagine. So the first one would be ASP. I got GAU, G, A, U, so they all intersect right here in aspartic acid, and UAC. Let's see, UAC, tyrosine. And so all that's doing is each three of these codes for one of these. So we call it a codon. Now there's a couple special codons. We have a start codon, which is only AUG. And that's because that's where translation always begins. And then we have, and it's right here by the way, and then we have stop codons. We have three of them, UAA, UAG, UGA, UA, UAG, and UGA. And what they do is they stop translation, because if you notice, there's no amino acid coded for. So what happens there is that tells the amino acid chain, you're done, get off, and to move on. Okay, so now we got some vocab down. Now we can kind of look at the processes. Okay, over here I have a picture. I highly recommend you either printing this picture off or finding a picture on the internet yourself of translation and printing it out so you have something to refer to. Okay, steps are, first thing that's going to happen is that the ribosome, remember the ribosome, little thing usually found in cells where proteins are made, is actually made out of two halves, a top half and a bottom half. So the small subunit is the bottom half, so that's this guy right there. And he's going to find the start codon, and it's kind of hard to see, but right there, actually, you know what, hold on. Let me see if I can make this bigger for you without getting, okay, wait, there. Now it's a little bit bigger for you to see. Okay, so the small unit right here is going to find the start codon. So he goes up and down the mRNA strand until he finds AUG. So he found AUG. Oh, light. Come on. Hang on. Okay. So he found AUG, so that tells him to glue himself on right there. And then an amino acid with tRNA right here, he's going to come down and sit on it. Now the only way that he can sit on it is if he fits with it. So if we start out with AUG, well then the tRNA needs to be opposite. Opposite of A is U, opposite of U is A, opposite of G is C. So the tRNA that has UAC on it is going to stick to it. We call that an anticodon. So if a codon is the 3 on the mRNA that says what an amino acid is, the UAC is the opposite anti to the codon. So here's AUG, here's UAC. So they can stick together. They can hydrogen bond together. So he's going to come along and he's going to bring along with him an amino acid. So he sits down, glues himself down on there. Then the large subunit, or the second half of the ribosome, is going to sit down and he's going to glue himself on it as well. And what I end up with is this structure right here. So I've got a whole entire ribosome, I've got my strand of mRNA in the middle, and then I've got my first tRNA sitting on the start codon ready to do its thing. Okay, next thing that's going to happen, oh, well, I kind of said this already, that the complex is built and ready to go. Okay, so I've got this guy, he's all sitting there looking good. So now we're going to do the next thing, which is to, oh, I have a text message. Okay, so now we're down here. So next thing that's going to happen is the ribosome has these 3 seats, and the first seat was already, or the middle seat is already filled up by that first tRNA. So the one to the right of it is open. If we look at, let me scoot that, you can see it's open right there. Okay, so it's open, and that invites the next RNA or tRNA to come down. Well, the codon is GUC, so I need the tRNA that's going to fit to that. G fits with C, U fits with A, C fits with G. So whichever tRNA has the anticodon CAG, he gets to sit down. So tRNAs come and go, but the one with CAG sits down, and he brings along with him the second amino acid. Okay, so next tRNA comes and attaches to the next codon onto the mRNA. After that, we need to pass off the amino acids. So let me again make this a little bit bigger so we can see it. Okay, so the first amino acid that was there, he's going to pop off and jump on top of the second one. And that's going to happen through a peptide bond, you can see right there. So, and we learned this back in chapter 2 again, that amino acids are held together in a protein by peptide bonds. So the first amino acid jumps off, attaches himself with a peptide bond to the second amino acids. So now he's empty. He's got nothing there. So the ribosome can't do anything else but scoot over. So he's going to scoot over one codon to the right. And what happens is that puts my first tRNA in the first position. So that's the kick out seat. So someone's in this seat, that means they're about to get kicked out. So now this guy is in the middle seat and I've got a new open spot. Well, that new open spot is UUC. So my codon says UUC. So my anticodon must be AAG. And so my tRNA that says AAG on his anticodon can come and sit down and bring over the next amino acid. So looking at this, the ribosome moves one codon to the right, kicking out the first tRNA, allowing the third tRNA to come and sit down. Okay, this is going to continue. Chain of amino acids continues to grow until a stop codon is reached. So this just keeps on going. Let me scoot this over. Let me scoot this guy out so you can see it better. But this process of scooting over, allowing the next one to sit, popping off this one, then he's going to scoot over again, allows the next one to sit, he pops off, scoot over again, next one sits down, he pops off. This process just keeps on going until I get this nice big long chain of amino acids. Once I hit the predetermined length of my amino acids, I am going to come across what's called a stop codon. A stop codon, one of the three that we mentioned, UGA, UAA, and UAC, these guys, no, hold on, I messed up. UAA, UGA, UAG. I said UAC, I meant UAG. What's going to happen there is that the tRNA is going to come, but he doesn't have any, I need to scoot this over too, he doesn't have any amino acids attached to him. So the tRNA comes along and attaches to the ribosome, but he doesn't bring an amino acid. Notice he has no amino acids. So he's going to come and sit down, but there's nothing for this guy to jump onto. And if there's nothing for him to attach to, he just bounces right off. So you can see this is 8 where he comes in like this, but when he jumps off, there's no one there to attach to. And so what happens is that the ribosome, the amino acid chain, and the tRNAs and the mRNA, well, they all just fall apart. They just all fall apart. And that allows them, whoops, I thought there was one more. That allows them to just take off and go do whatever they're going to do. So here's that last tRNA. There's the tRNA with no amino acid attached. Here's the 2 halves of my ribosome. There's my mRNA. And there's my completed protein. Now, if you remember, the Golgi apparatus is the space where proteins get packaged and processed and dealt with. So if you can imagine that this guy, his job is now to go to the Golgi apparatus to get spiffed up for export out of the cell. So, kind of confusing. Yes, I understand. It does take a lot. So I would go back and watch this maybe once or twice. I'm sorry, it's 14 minutes, but it's good to watch a few times. Also, again, watch some animations, do that active art in the textbook to help you understand this, because it does take a little while to get going. Okay, if you have any questions for me, ask me tomorrow. I'll talk to you later. Bye.